All right, in this lesson, we are learning about genetic modification and cloning. This is topic 3.5, but remember, it is only about half the topic. 3.5 dot points. Genetic modification and cloning are covered in this lesson only. All right, we're going to talk a lot about genetic modification, but I want you to know the difference between genetic modification and selective breeding. Okay, selective breeding is not genetic modification. Genetic modification is taking or altering a genome by taking another gene from another species and implanting it into that genome, right? Completely different species mixing genomes there. But this is really different to selective breeding. It's breeding to keep particular beneficial traits in a population. So, for example, carrots are a really classic example of this. They are initially purple, right? That's their natural color, but they were grown in Holland and the Dutch love the color orange and they wanted to make more orange ones to satisfy the king. So they bred more and more of those orange ones just by taking the mutant version and just breeding those, okay? Wild carrots also can be skinny and pale. So there's so many different versions of these, but it's about taking the ones that you want and growing those based on the beneficial characteristic that you're looking for. So if we only take two really tall people out of a population and breed them and start a new society off them, then there might be more chance that the entire society is quite tall. Now, selective breeding is used in pretty much every single agricultural industry, and everything that is a crop that you eat has probably been subject to this. There's evidence to suggest, and questionable news sources here, that chickens are bigger these days, and we're talking without hormones. There haven't been hormones used in chickens for over 40 years in Australia, so it's more about just selecting those larger ones and breeding them so that commercially there's more meat available. Same thing with eggs. Apparently, I don't know, can't confirm that eggs today are able to produce more, chickens or hens these days are able to produce more eggs compared to many years ago. Modern corn also has a very different look to it than what it initially started out as. So we're talking about taking mutant varieties or, mutations can be beneficial and changing the phenotype and therefore only breeding those. And we can talk about domesticated dogs. They've been selectively bred for hundreds of years. We all have many different subspecies of them, but they are all from the same species. It just depends on, if you've bred different behaviors or body shapes for different purposes. You wouldn't compare a husky and a sausage dog, for example. They're doing different jobs. All right, so genetic engineering is very different from the selective breeding we're talking about. This is where we start talking about actually moving genes from one species into another. Now, this can occur because DNA is a universal code. It can be read regardless of the organism. Every organism uses that kind of DNA code. So transcription and translation of an implanted gene can still happen because it all works through the same process. And this really is a great example. Insulin production is completed using bacteria by taking a gene of the insulin from, say, humans, implanting it into a plasmid of DNA in a bacterial cell, and then they get that transcription translation going and insulin is produced. Lots of different things can be genetically engineered. So spider silk protein can be produced in goat's milk in order to commercially be used as a really strong fiber. It's much quicker and much more reliable to have it harvested this way than just harvesting spider webs. Purple tomatoes have been created using genes from snapdragons, and it darkens the color, puts in more antioxidants into the fruit, and they have the nice cancer fighting properties. And golden rice is a fantastic example of this. Now, rice is a mainstay for all of so many cultures as the main staple of their diet, but it doesn't have a lot of nutrients in it. So scientists were able to take a beta-carotene gene from corn and also bacterium and implant that into a rice genome, and they produced rice that has an increase of vitamin A in it. So that's really helpful. All right, gene transfer. This is the beautiful BioNinja version of the insulin production. It uses enzymes. We love enzymes. And restriction enzymes are used to cut out the relevant gene. And it's also used to open up the genome of the bacterium. And it just leaves a little sticky end so that they can all stick once that new DNA is implanted. The genes are then transferred from one organism into the bacterial plasmid. And at that stage, it's called recombinant DNA, or recombinant plasmid in this case. It's a combination of the new gene and the old bacterial genome, and it's put back into the host cell. At that point, it's called a transgenic organism, all right? It's got genes from different species, transgenic. And then DNA ligase, another enzyme, it joins up the backbone, that sugar phosphate backbone, and makes sure everything is sealed properly. And then you can go and grow those cells in culture and extract the insulin protein that has been gone through transcription and translation. So obviously there's some ethical concerns around genetic modification, and you probably already heard them, but we talk about, playing God. And there's a bit of a cost-benefit analysis that has to come from creating these organisms. But it's really hard to find reliable, independent, unbiased data on the internet. Okay, so each country is kind of in control of their own control and regulations. So you might be talking about having specific labels on foods that are GMO. There's quite a lot of protests and people who just flat out disagree with it. And it's possible that the technology grows faster than the regulations. So there's many, many concerns around the safety to farm and the safety to eat and things like that. And not everyone's an expert, sadly. You wouldn't know it on the internet, though. So this is your BioNinja picture, and they sum it up really nicely for you, but I want you to understand that there are so many opinions out there, and they all need to be backed with scientific evidence, not just fear-mongering. So from an international perspective, we might not be considering things that are considered benefits for them. So different cultural practices in different countries, you might be talking about different climates or changing climates. There are cultural practices that rely on the land and farming that don't necessarily mean the same to us as they do to them. They don't have the same opportunities to rock on down to Woolies and buy some packaged food. So once again, some risks and analysis. We're talking, there's a fantastic example. Early on there was concerns that a GM corn crop that was bred to contain a particular sort of toxin that a bacteria would make, and it stopped requiring pesticides to be used on this crop. But they were saying a butterfly population that would land on a nearby plant would have that plant pollinated by the corn, and therefore their new plant would produce that toxin, and the butterflies were dying off. But the study was repeated, and they found that there's no significant difference between the populations near the GM corn and the other fields, so it was completely debunked. But it's really important to know that all this stuff relies on scientific reliability, repeatability. Otherwise, stuff can become, or the facts can become lost in sensationalist news and panic. Okay, let's talk about cloning. So cloning is the production of genetically identical organisms. Now, twins are technically clones. You've got genetically identical organisms there. So twins, triplets, all that kind of thing, they are technically clones. And cloning can be found in nature in many, many places, but it's often associated with an artificial cloning technique which produced this lovely sheep known as Dolly. Every asexual reproduction type is essentially cloning. So we're talking mitosis and binary fission in bacteria. Garlic bulbs all originate from the single bulb, and strawberry plants put out runners from their parent plant and create genetically identical daughter plants. It's also budding from hydra, fragmentation from starfish, and spore formation from particular plants. When we start talking about intervention cloning, then we have different things to talk about. So animal embryo splitting is one version of this. Now we can create or fertilize an egg with a sperm, but once that zygote starts to split and divide and go through mitosis, it gets to about an 8-cell stage, and we can take one of those cells, and they are pluripotential. They have the potential to become anything. So they can be taken away and implanted into a surrogate parent. So we're talking surrogate, like all these cows here are now gestating the same cow baby or the same calf. But the disadvantage of this method to produce a number of clones is that you actually don't know at the zygote stage what kind of phenotypic features you are going to get. You don't know whether that cow is going to be very muscly or very small or, diseased or something like that. So not the most efficient way of doing things. Another way to do it is through somatic cell nuclear transfer. Now, this has an advantage. What we're doing is taking adult stem cells, adult cells from any grown organism, and you know what phenotypic features you're getting. Hey, look, that person's really fast runner. Let's take a cell from them and create a clone of them. The cells have already differentiated though, so you can't just make a new organism straight out of that. So what we do is we take an egg cell, we remove its nucleus, we take the diploid nucleus from our somatic normal cell from that speedy person, and we put that diploid nucleus into the egg cell, and we allow it to go through mitosis. That way, you actually know what features you are going to get, and that's what you're going to get at the other side once that organism has been grown. Now, this has therapeutic uses. It's not just about creating an entire new organism. You can create specific tissue types. So you might be creating pancreatic cells to replace in someone who's got type 1 diabetes and doesn't make insulin, or you could be regrowing nerve cells in a patient with spinal damage. So therapeutic stem cell use like this, or therapeutic cloning, has different uses than, say, just making a brand new sheep because we can. All right, once again, only about half the dot points, but quite a lot in this particular lesson.